It's a great honour and also actually rather intimidating to have to give a talk. I've only got around 30 minutes, and I really wasn't quite sure what to say. Something that to epitomise and work through my career to date, tell you a lot of things that maybe I've told you before, talk a bit about myself, maybe not, or just talk about the very newest data. So what I finally decided might be appropriate is to just give you a very quick rundown in a minute or so, a little bit about myself, move on to seg into Geoffrey Harris, because I think not everybody knows exactly who or what he did. Then talk about one of the aspects of my career, which is looking at pituitary tumours and how they form. And then finally, I hope, most importantly, try and see where this whole area and field is going into the future. And those of you who've come to find out what on earth the title means, you'll have to wait to the end, so don't leave yet, okay? I know that's the reason most of you have come. Okay, well, we'll just start off. I'm really delighted to be here in Poland. In fact, my family come from this part of the world, from Lodz and from Białystok on my father's side. But of course, in those days, all of this was actually Russia, and also from the Ukraine, and who knows what that may be tomorrow. But at that time, Wrocław was actually in what was then Germany, and it was called Breslau. And I suppose it shows the fluidity of the boundaries of Europe, but we are all indeed a European nation. And in those days, this was in fact Breslau, now Wrocław, which has clearly changed a great deal since then. Well, of course, I myself was born in the UK, in spite of this Eastern background, and had spent a little while at university, about 10 years in fact. I started off in Cambridge, where I started on natural sciences, moved to London, where I read psychology, then on to Birkbeck College, where I read social anthropology as part of my psychology degree, and then still not quite decided what to do, went to University College and read neurosciences for a degree, but then finally decided, probably I spent long enough dithering and decided to go into medicine, where I was at University College Hospital Medical School. In fact, at that time, I then decided probably I would become a neurologist, but this new area of neuroendocrinology did sound rather interesting. So I decided to go for 6 months to St. Bartholomew's Hospital. Well, I stayed rather longer. This is a hospital as it was when it was originally built in the main part. In fact, it doesn't look terribly different today. But what is rather nice, as many of you who know the hospital, is it has most magnificent 18th century buildings. This is the Great Hall at Barts, which is still a most magnificent place, and was only kept alive because of the King Henry VIII at the time. So, more recently, having spent a great deal of my career at Barts for some 30 years or so, some 3 years or so, I moved off to Oxford, 3.5 years or so. This is the Oxford Centre for Diabetes, Endocrinology and Metabolism. And I was quite intrigued on the first day or so to find our inpatient ward was actually called the Geoffrey Harris ward. Now clearly there was a reason for that, and that's that Geoffrey Harris spent most of his active career actually in Oxford. So if you go to South Parks Road, which is part of Oxford in the University in the Department of Anatomy, you'll find the Geoffrey Harris Laboratory, which in fact still exists. It's slightly updated, but frankly, not a great deal and not very different to what it was in the 1950s. But these are still active laboratories and now run by another group of very talented people. But then, who was Geoffrey Harris? Well, he was a man who very much maintained that experiments, not morphological description, were the key to the demonstration of the hypothalamus and the pituitary, even though he was a professor of anatomy. He was born in 1913, spent his early career first at University College London and then Emmanuel College, Cambridge. In those days, to be a professor of anatomy, you had to be a clinician. So he then travelled on to St Mary's Hospital, moved back to Cambridge as a lecturer, then moved back to London, which was the Maudsley Hospital, which is actually an institute of psychiatry and is now part of King's College London. And it's as interesting as I end my talk, you'll see we've gone round in a big circle. But in fact, he spent the majority of his active professional career as professor in Oxford and fellow of Hertford College. What did he actually do? Well, he found when he was an undergraduate, he was intrigued by this question, that if you have seasonal breeding animals, then clearly you have animals responding to cues in the environment which are changing the activity of their hormones. And by that simple insight or observation, this made him think that environmental stimuli acting through the nervous system could lead to the release of what later became known as the releasing factors from the hypothalamus. These would then go through the portal system of blood vessels, so he showed there, by analogy with the hepatic portal circulation, and these will then link the undersurface of the hypothalamus and control the pituitary. And although this seems obvious to us now, at the time it was highly controversial, and he got a great deal of really rather vitriolic flack from a lot of people. But in fact, his very careful experiments clearly demonstrated this. In fact, they were rather interesting. Here you'll see early demonstration of the portal blood vessels, and I showed the anterior chamber of the eye, because what he actually did in some of his experiments was to transplant the ovary to the eye and then show how you could alter the morphology and ovulation by changes to the hypothalamus and pituitary. And everything that we now know in neuroendocrinology flows on from those very early, seminal and very elegant studies. He died actually in 1971, and he's now buried in Bladen Church, which is about 7 or 8 miles just north of Oxford. I tried to go there recently, but the weather's always been so bad, I've never quite managed it. But that is where he was buried. In fact, if he had lived a little while longer, I'm quite certain he would also have shared the Nobel Prize with Grossman and Chalet for the discovery of the way that the hypothalamus communicates with the pituitary. So let's go on to the meat of what I was going to talk about. And one aspect of my career has been very much interested in where do pituitary tumours come from. Now, we know they're common. We know in an autopsy series, it's around about a fifth can have very tiny tumours. But we also know from really nice epidemiological studies from Albert Beckers in Liège and from Nikki Karavataki and John Moss in Oxford that actually they're not uncommon. It's 1 in 1,000 prevalence in the general population. And this is not just in the Liège Valley, which we originally thought when we saw Albert's work, but it's everywhere. So we're dealing with a common problem. And if we're dealing with that, we have to think, how can we work out where they come from? And one of the easiest ways of developing it in this world of molecular biology is have a look at genetic syndromes. See the syndromes in which pituitary tumours are a prominent feature, and then if we can find those genes, we can feed back to sporadic tumours as well. And you'll all be aware that there are a lot of genetic syndromes associated with pituitary tumours. These are particularly somatotroph, but all tumours. The MENs 1, 2, 3, and now 4, Carney complex, McCune Albright, the SDH syndromes, the new kids on the block, and FIPA. And it's been remarkable over the time I've been an endocrinologist that the major genes belying all these syndromes have been discovered. And just as an aside, I'm not going to deal with the SDH syndromes. These are the newest and very interesting, but very little studied. Neither am I going to talk about FIPA and AIP, both because Albert Beckers and Marta Corbinets could do this much better, but plus the fact that somatic mutations have not been seen in AIP in pituitary tumours. So let's just have a look at some of the other tumour syndromes and see whether they can teach us something about sporadic tumours. Well, very quickly, the MEN1 associated tumours you all know, pituitary tumours in about 30%, most are prolactinomas, but it's very rare to find somatic mutations in sporadic tumours in the menin gene. We looked at the expression of the mRNA many years ago, and that too was absolutely normal in pituitary tumours of a sporadic variety. So we're not really learning much from that. And even now, menin and its precise function and mechanism remains rather unclear. In the MEN2 and 3 syndromes, it's quite rare to get pituitary tumours, or you do see them. And very recently, one of my registrars published a paper showing Cushing's disease could be associated with a genetically proven MEN2. Now, we know from the earlier work, and I'm delighted to see Anna Spada here, and the absolutely seminal work on the GS-alpha mutations, that these occur in sporadic, around 30% of somatotroph tumours. One of the more interesting recent pieces of data coming from those, of which we had a small collaborative part, was the realization that actually GNA-alpha is imprinted, maternally imprinted in pituitary tumours. So if you look at which allele the mutated gene alpha comes from in the sporadic tumour, it's almost always the maternal allele, because the paternal allele is not generally expressed. However, once the tumour forms, then the imprinting is relaxed, and both alleles are then expressed. But it still remains a little unclear as to the involvement of this pathway in sporadic tumours. Clearly, these mutations are not a necessary condition for all tumours, and it still remains a little unclear as to whether they are a sufficient condition for the average tumour. But in a not unrelated area, in the Carney complex, we see here patients who have these macules and strange facial features. We have atrial myxomas, we have nodular adrenal disease, and in particular in Carney complex, about 10% have symptomatic acromegaly. And we know from the work of Konstantin Stratakis that in the majority of patients, you have a germline mutation of protein kinase A regulatory subunit. So we thought it would be quite interesting then to explore this in sporadic tumours. So we have, looking at the very simple pathway, GHRH going through adenylyl cyclase, and we've got the GS alpha subunit, remember. Cyclic AMP is released. This interacts with the regulatory subunit, and that then allows the catalytic subunit to activate CREB and phosphorylate it and cause growth hormone secretion and thereafter proliferation as well. So clearly it sounded quite a nice idea that sporadic somatotroph tumours would also show mutations of the PRKA subunit. But sadly, in our studies and in Konstantin Stratakis' studies, this was never found. It was a good idea, but it didn't quite happen. What we didn't think at the time is anything about the catalytic subunit. So if we now go back and think about Carney complex then, let's just think for a moment about the catalytic subunit. As I said, this we thought was not going to be directly involved, but we do know from some very elegant work from Felix Birchlein and Brulio Lolio that this may be an important integral part of tumor regenesis. And in some very nice work that I'm sure many of you know, they looked at adrenal adenomas from patients with Cushing's syndrome. So if you like, a nice model for acromegaly, small tumors, generally benign, which cause hormonal secretion. And in some very lovely exome sequencing, they found that just over a third actually had mutations in the protein kinase A catalytic subunit A. Now, I'll show you in just a moment, but it was almost always the same mutation at codon 206. And what happens, as I'll show you, is that this causes constitutive activation. And shortly thereafter, the Stratakis group then also found that in patients with acromegaly, and that there was a germline syndrome where there was a duplication of protein kinase A catalytic subunit B, which is a very similar isoform. So in both these situations, there seemed to be an association either with tumorigenesis, and particularly in this instance from Stratakis' work, with growth hormone secretion. And just to show you what they found is that at this particular position was very much concerned over evolution. There was a somatic mutation within the tumor compared to the normal, and that at this particular site, the PI site, this interaction that normally occurred was blocked structurally when there was this mutation. And I know that there was some other work that they are planning on submitting at the moment, which looks at the structural biology of why this would occur. But essentially it means that it cannot be blocked by cyclic AMP, and therefore there is constitutive activity. Well, this was again a great candidate, and in the last couple of weeks, we've looked at a whole group of somatotroph sporadic tumours, both from Oxford and with Marta Corbinitz from Bart's. And sadly, and yet again, we found that none has shown either the catalytic unit subunit A or B mutation. So it's really been quite frustrating, as you can see, over the years, looking for candidate genes and not really getting anywhere. We can look at other aspects of the signaling pathway in the somatotroph cell. We can look at the GHRH receptor. Is it constitutively activated? The answer is no. The adenylyl cyclase pathway we've talked about. You can look at the IGF-1 and the growth hormone receptor. We found no mutations in the growth hormone receptor. This was subsequently denied by another group, but then in more recent studies, we've confirmed that in all types of somatotroph tumor, the growth hormone receptor is not mutated. There may be changes in expression, but not in actual sequence. Other groups, including Stefan Peterson, have looked at, for example, the somatostatin receptor and found this is very rarely mutated. It looked at the ghrelin receptor, very rarely mutated. So it's really become quite frustrating. Well, how about maybe a smaller tumor, one which we can examine very carefully, which we know is very small and therefore might reveal the secrets of tumorigenesis more easily? What about Cushing's disease? Well, I'm afraid, and I won't go through all the data in detail, it's been equally frustrating. Originally, people looked for activating mutations of the CRH receptor and found none. We and the French group with Betania also looked at the vasopressin receptor and found it to be normal. And the glucocorticoid receptor very, very rarely is mutated, except in very occasional patients with Nelson syndrome. There were even detailed investigation of the pathway whereby cortisol interacts with the glucocorticoid receptor through the HSD enzymes. And while there are changes in these enzymes, they don't seem to account for the actual tumorigenesis process. Again, the ACTH receptor is expressed on corticotrophs, but it's not mutated. And again, changes of expression, I believe, are secondary to and not causative of the tumor itself. And lastly, remember, we mentioned MEM4. Now, this was actually a syndrome based on MEN-X, which Dr. Pelagata very elegantly showed first in the mouse. And it's a syndrome of P27 germline mutations. And you can get a variety of different tumors in this syndrome. But way back in '98, Patricia Dahia looked at P27 in sporadic tumors and found it was not mutated. And indeed, when with Marta Korbinitz, she looked at the MEN1 negative germline syndrome. So these are patients who appear to have MEN1, but no MEN1 mutations. She, and in fact other groups, were unable to find any mutations in these patients. So P27 may be involved, as I'll show you, but it's not in terms of its mutation status or its mRNA. And in fact, what she did show many years ago with us is that if you look at normal tissue and the amount of P27 protein, very carefully quantitating immunocytochemistry, you could show a lower amount of P27 in every type of tumor, particularly corticotroph tumors. And what we therefore showed was that P27 protein was diminished, but P27 mRNA expression was normal and was not mutated. So this led us really inside the cell in terms of the cell cycle. Now, many of you are aware that the cell goes through the cycle, cyclin D, cyclin E, and then it progresses through. There's a phosphorylation of retinoblastoma, and remember, knockouts of retinoblastoma in mice cause pituitary tumors, and then cycle progression. But these are in turn regulated by breaks, if you like, at P16 and P27. P16 I'll come back to, but P27 is regulated by protein degradation. So how do you control the protein? So P27 is low, and certainly in corticotroph tumors, cyclin E is high. Now there's a yin and yang between these two. Low P27 increases cyclin E, high cyclin E lowers P27. So what we tried to do was explore what is it that controls the protein degradation of P27 and cyclin E. And we looked at the pathway of P27 degradation, which was very interesting. P27 is ubiquitinated by a ligase, and this complex then combines with it, but it will only interact with an F-box protein, which is the one that recognizes it, as in a jigsaw puzzle. And this lock and key is controlled by so-called Skp2. So we argued, if you have low P27, then it must be actively degraded, and Skp2 was going to be increased in level. Anyway, Madeleine Musat looked at that when she was with us, and in fact the mRNA was absolutely unchanged for Skp2. There were subtle changes in the protein which were quite interesting, but not enough to actually account for the p27 degradation. Okay, so we looked at the other end. What about cyclin E? Cyclin E is ubiquitinated in a similar way, but here the recognition protein is different. It's called archipelago. So the argument here was, well, maybe if you have very low levels of archipelago, you'll get high levels of cyclin E, more degradation of p27, and that is the causative factor. But again, we were wrong. And if anything, AGO, or archipelago, is overexpressed, maybe as a compensation, in pituitary tumours. So we were left with this conundrum of what is it that causes low p27? And in fact, what came to our help was some recent data which showed that a protein serine kinase called AKT would specifically phosphorylate p27 and enable it to be degraded. And lo and behold, for the first time we got positive data. Many of you will know the results that AKT is overexpressed in pituitary tumours. So here is a normal pituitary, and the immunocytochemistry clearly shows it's overexpressed in non-functioning and other tumours. And what we believe therefore happens is that this PI3 kinase AKT pathway is overactive, phosphorylates p27. This blocks the import into the nucleus, which allows the cytoplasmic degradation pathway to occur and lower the levels of p27. So you can see at each stage we're pushing back the locus of change that we've been investigating. There's another parallel signaling pathway to the PI3 kinase AKT, and that's one again, most of you will know, the MAP kinase pathway, which again, from the growth factor receptor through BRAF, which is pivotal, causes proliferation and changes in gene expression. And again, we were very lucky. We had with a medical student amongst us at the time, finding that the BRAF mRNA was overexpressed in a group of pituitary tumours. It was not mutated, as it is in melanomas, as I'll show you in some other tumours, but it was overexpressed. That allowed us actually to proceed further when Dorota Drokowska, who is in fact Polish and was working with us at the time, also looked at the other aspects of the MAP kinase pathway. And she again found that the phosphorylated ERK, which is the last part of the MAP kinase pathway, phosphorylated ERK is overexpressed compared to ERK being overactive, and that was generally true for all pituitary tumours. And to really summarize then where we got to at that stage about 4 to 5 years ago was that we could show that BRAF, MEK, ERK in the MAP kinase pathway and AKT in the parallel pathway were all overexpressed. And these are all pro-proliferative. And that in turn could cause changes in p27, although there are other factors that can do that, and Marilu Therivopoulou has shown other mechanisms for p27 degradation and also changes in c-MYC. But if you look really downstream, you don't see any changes. mTOR, cyclin D, S6 kinase, it's as though proliferation occurs up to this point, but then something is blocking further progression into the proliferation stage. Well, there is one possible candidate, and that's senescence. Senescence is a process whereby at a certain number of proliferative occurrences, the cell stops dividing. So we looked for senescence markers, and Kristalina Alexandraki was with us at the time and found that in growth hormone tumours and in non-functioning tumours, there was in fact a slight increase in senescence. I don't believe this is the actual cause of the fact that these tumours stop proliferating, because there is an absence of senescence in prolactin and corticotroph tumours. But it is interesting that when you look for pituitary carcinomas, they are almost always occurring in cells which derive from the prolactin or ACTH lineage. And we're just speculating, and it's no more than that the absence of malignancy in the majority of growth hormone and non-functioning tumours is due to some senescence, and the tendency for the prolactin and ACTH tumours to become malignant may be due to its absence. But I don't think that's the whole story. But where we've come to, really, is to, in fact, conclude there are a whole variety of oncogenes and tumour suppressor genes that are altered in pituitary tumours. If you like, it's a form of stamp collecting, it's a form of description showing that retinoblastoma, P16, P18, etc., all of these appear to be controlled or downregulated by methylation of their promoters, and the original studies by Bill Farrell clearly showed that. With a P27, MEK and ERK, there are changes in the protein rather than the mRNA. But what you see here is a program, as it were, of proliferation. But none of this program appears to be the primum movens, the originating cause of the tumour. So where is this locus of mutation? What's going on? So I'd just like to stop for a moment and just think theoretically, just consider, do a mind experiment, a Gedanke experiment as to how tumours form. Let's think a little bit more basically. Now, we know with Vogelstein, when talking about pure cancer, and this is a log growth curve, he's shown, and in many other cancers like breast cancer, there's a series of progressive mutations, each one becoming more malignant than the one before. Now, we know pituitary tumours probably only have one or two mutations and have a uniform growth curve. So really, we would expect, with a log growth, one mutation and the tumour to look just like this. So let's go further and do some calculations. Let's just take a possible non-functioning pituitary tumour. Let's assume, for simplicity, it's a sphere. Volume of a sphere is 403 pi r cubed. Let's say it's 1 centimetre in diameter, and therefore we've got a tumour volume, on average. Let's think about the individual cells. Each cell is going to be, on average, about 20 microM in diameter. Again, you can therefore work out, theoretically, the volume of a given cell, which is 4 times 10 to the minus 12 cubic centimetres. And it's very simple then. So how many cells in the average tumour? So it's total volume over number of cells over the volume of each cell, and it's round about, give or take, 10 to the 12, 1 trillion. So that's our number of cells, and that's the one that we see in a non-functioning tumour. And we believe that the tumour has grown, we'll assume, from a mutation, and it's grown exponentially. So there it goes, gets bigger and bigger, and then it presents itself. Well, we need to know the doubling time to work out how long that's been there. So let's work out the doubling time, and there are a number of estimates, and they're pulled together from different studies. And Chen recently in Pituitary just worked out the average doubling time is about 3.5 years, give or take. So we can put that into the equation and see when the tumour started. So the tumour contains 10 to the 12 cells, assuming uniform exponential growth. There's probably 40 multiplications. What do we get to? Well, the tumour started at about 156 years, minus, which is a little bit problematic at this stage and leads to certain problems. So what's what a Latinist would call a reductio ad absurdum. So our premises were wrong. And in fact, we know they're wrong, because we know that many tumours don't do anything at all. In this study by Nikki Karavataki and other studies have shown similar results. She looked at 24 macroadenomas, no intervention, 50% did not grow. So clearly we're dealing with a different animal here. So what I'd like to suggest is that rather than looking for these growth curves for your typical cancer, we're looking at something more like this. A tumour that grows, maybe with one mutation, reaches a plateau and then stops. And actually we know that there are tumours like this, for example, in the parathyroid, and Andy Arnold has shown that when you get a translocation where cyclin D is driven by a PTH promoter, you can get exactly that. You shift the dose-response curve, the tumour reaches a new steady state, and we all know many of our hyperparathyroid patients don't change for 10 years or more. So I think at this stage, what I was assuming is that, let's say the tumours arise from monoclonal mutation. This is counteracted by an opposing force at a given size, and there may be something to do with cell contact or membrane-associated states. And both Jacqueline Tuyas and Gunther Staller in his group have shown how these interactions may occur. So that, theoretically, then, is maybe what's going on. One mutation, it grows, it reaches a new state, it stops. The problem is, we still have not identified what this mutation is. Let's take a completely different approach. And with my colleague Raj Thacker in Oxford, we decided we'd look at a different way of doing it, and Paul Newey is here and very elegantly carried out this study using total exome sequencing. So what Paul did is take some very pure tumor samples and germline leukocytes and use a test set of 7 paired samples. He would then do exome capture and sequencing, variant calling, and he'd validate it against a second validation set of 31 tumors. Easy. Look for the changes, and remember, this is the sort of thing which Felix Beutschlein and Bruno Allolio were doing for adrenal tumors, and many people have done for other tumors. So really exciting. What happened? Well, 21 out of 24 mutations occurred as single nucleotide variants. Some of them were small insertions of deletions. But the 24 somatic variants occurred in independent genes. There were no variants in genes previously associated with pituitary tumors. And when you use protein prediction software as the so-called driver mutations, these could be confirmed when we use direct sequencing, but no specific mutation occurred in more than one tumor. If you like, very nicely done by Paul, but deeply depressing, because the answer did not occur. So here we are in 2013, burgeoning into the 21st century, having the first idea what's going on. But I think help is at hand. And in the last part of my talk, I'd just like to suggest a possible different approach or a different way of looking at this type of problem. And just would like to remind you of another signaling pathway, which I'm going to discuss, which is the Wnt-catenin. Now, in the Wnt-catenin pathway, beta-catenin is bound in this complex, which phosphorylates it and it is degraded. If you get a Wnt ligand, this interacts with the so-called frizzled receptor. That dissociates this whole complex, and this allows beta-catenin to cause proliferation. So the critical point is that there is a sequence of beta-catenin which allows it to be phosphorylated and degraded. And it has now been shown that in many tumors, this particular sequence of codons can be mutated, stabilize beta-catenin, and allow proliferation. So beta-catenin, if you like, when mutated, is an oncogene. Right, if you're still with me, we will just now move sideways before we move back. And the sideways move is a craniopharyngioma. Horrible tumors, very difficult to treat. Two major types, the adamantinomatous type and the papillary. And some years ago, it was clearly shown that the adamantinomatous group frequently show beta-catenin mutations. But the beta-catenin is only translocated in a small cluster of cells. And these are very strange cells. And these studies, and I'm very grateful to my colleagues for providing some of these slides, these beautiful studies by so-called JP Martinez Barbera, Carlos Gaston Masway, and Cynthia Antoniadou, all at University College Hospital originally, although some of them have now moved on. But I think the theoretical arguing behind these very elegant studies might provide a solution. Because what they then did is take pouch precursors, and these were going to be stem cells, put in beta-catenin in its mutated form, and show that you would cause tumors. And you can see that these tumors, where the beta-catenin is translocated, are very similar to what you see in human craniopharyngiomas. We too now have very similar data showing the situation in human craniopharyngiomas. If you just follow this through in his most recent work, the fascinating point was that when these mice developed craniopharyngiomas with the beta-catenin transgenically put into stem cells, you could clearly see the tumors, you could clearly see the beta-catenin around the tumors, but the tumors themselves, shown with the asterisks, were negative for green fluorescent protein. They were not from the mutated stem cells. So this shows a completely different possible model of tumorigenesis. And so what JP has actually suggested, and we're now working together to develop this, is that within the stem cell, there is what it's called non-stem cell autonomous tumor formation. You have these clusters of stem cells. These cause various changes in the surrounding stroma, so-called inductive signals, and at some point, one of these may then mutate in a so-called tumor propagating cell, which is not a stem cell. It's separate from that. So what I would like to say is that maybe this type of process is going on, and it doesn't have to be beta-catenin. But I mentioned to you the adenomatous craniopharyngiomas show beta-catenin mutations in these cell clusters. And at the end of last year, a group from Harvard looked in more detail at the papillary craniopharyngiomas, and around the same time, slightly delayed, sadly, we also showed that the papillary craniopharyngiomas almost all show BRAF mutations. So you can change a variety of different signaling pathways in the craniopharyngioma and then cause secondary tumors. So I think this is rather exciting because it suggests it may be a very different model of what's going on. So I'd just like to suggest, finally, another hypothesis, that the tumors arise from stem cells, and remember this is a speculation for pituitary tumors, but let's suggest that tumors arise from stem cells which secondarily cause this program of proliferation I've discussed before of the surrounding cells, and there may then be occasional random mutations, and it can be any proliferative gene. It may not be one gene that occurs in every tumor, as I show from Paul Newey's work. And there may in turn then be growth inhibitory modulators that stop the tumor going beyond a certain size. And if you then think of the problem in that way, you can start seeing all sorts of connections and other data that fit in so nicely. For example, in data from Bill Farrell and Dick Clayton many years ago now, when they were looking at Cushing's tumors, and we've all ignored it, well, these were not monoclonal, they were oligoclonal. There wasn't one single clonal entity. And a really beautiful, elegant study published a week or two back in Nature, these people took osteoblasts, put in a beta-catenin mutation, the osteoblast would hone into the bone marrow, and these mice then developed acute myeloid leukemia, which was different from the osteoblast cells. So it's a secondary formation. And this also works in very well between the interaction between cell and stroma, again from Gunther Staller's group. Many ependymomas, medulloblastomas, neuroblastomas fail to show recurrently mutated genes, just like we have found. If you take a transgenic mouse, where you have SOX2 nestin cells, stem cells, you can mutate them, and the melanotroph tumors are nestin negative. Again, these are secondarily formed around the stem cell tumors. And lastly, I think Clara Alvarez has published a great deal on stem cells, and I think is going to be talking tomorrow or the day after on the identification and isolation of stem cells from the pituitary. So, the one you've all been waiting for, what on earth am I talking about? For many years, I have to say one of my favorite books has been that written by Omar Khayyam. Omar Khayyam, as some of you have Googled and identified, was a mathematician, scientist, and particularly a Sufi mystic. And in quatrains in the Rubaiyat, which was beautifully translated by Fitzgerald in the century before last, the 19th century, I think have always summarized my philosophy of life in many ways. And I do advise you, in whatever language, to read it if you can, because it is a beautiful, world-encompassing and total philosophy. But in one particular quatrain, he identifies something which I think is quite interesting. And I'm sure you all sort of realize what this is. But for those of you whose Farsi is possibly a little rusty at present, let me tell you, the model is that there is a creator, and by creator, you may or may not imagine a person. It can be creation. And he said, all of us are like pots and we're being formed. And suddenly somebody shouted out, you know, where are you going? How are you going to form? And it said, none answered this. These are the pots speaking. None answered this, but after a silence spake a vessel of more ungainly make. They sneer at me for leaning all awry. What? Did the hand then of the potter shake? Did God make a problem there? Did he form him wrongly? Or was there an initial problem in very early embryogenesis? Are the cells that we're seeing wrong in cancer actual stem cells that have been there all around, all along, and may even have disappeared by the time that the tumor is actually seen? So what we're seeing is something that has occurred very late, but depends on something that maybe you're even born with. Maybe not mine as 150 years, but maybe in the very first initial embryogenesis. So you can all leave now so you understand where that came from, but since we're somewhere in the Middle East, I'll come back to that. So I'd just like to suggest now, towards the end of my career, where are we going forward? Well, I think the identification of stem cells within the pituitary and pituitary tumors using specific markers is a very exciting area in which to develop. I think we need to reassess clonality. Remember, most of this work was done with rather poor tools some 20 or so years ago. And I think we'll be looking more for oligo or multiclonality if we really look carefully with better techniques. And remember, the great majority of cancers now, we realize, are very heterogeneous. But I'm not saying we shouldn't find a specific mutation, a specific tumor. I think we'll keep looking, but I think these are going to be parallel approaches for the future. So then finally, just to say, as a sort of end of my, I won't say the end of my career, certainly the end of my talk, and just to return once more to the Middle East, there's another way what one can epitomize what one has been doing over the years. And from this part of the Talmud, of the Hebraic works, it's a very nice, not exactly a psalm, but a paragraph which always very much appealed to me. The day is short, the work is great, but the reward is much. It's not your duty to complete the work, but neither are you free to desist from it. So it's great fun. You can have a whale of a time. Hopefully, in the end, our patients will benefit from this type of research. I think there are lots more things to do in the future, and we all have an obligation to continue thereafter. So finally, as you can see, we've had amazing collaborations back at Bart's with a huge number of people. You can't read their names, that's done on purpose. There are just too many of them from every single part of the world, and it's been a great joy working with all these collaborators at Bart's. And then more recently, I've had the privilege of working with some of my colleagues in Oxford, which has also been great fun. And I'd like to point out to you, maybe the first announcement, that Prof. Thacker has now been made a Fellow of the Royal Society, which is an honour which is actually bestowed on very few clinicians. And I'm delighted that it's actually come to a clinical endocrinologist. So finally, I'd particularly like to thank Marta, who's here, and has always been a delight to work with. She often says she understands my brain more than anybody else, and that's probably true, at least the cortex part of it. And I'd like to also pay thanks to a great friend of mine and colleague, Rolf Gerhard, who was president of the NEA and sadly died some 3 years ago. Well, anyway, thank you for listening. Thank you. I hope you understand who Omar Khayyam was now, and thank you once again.